Well, no mai haramai. Welcome everyone. I think we'll kick off. We've given everyone a chance to join. Welcome everyone to Rangahau Rising. My name's Nick Waipara. I'm just going to open our hui today with a karakia to set our journey on the way and have hear from our wonderful speaker. So he karakia timatanga me te whakakapi kaupapa. Kia tau ngā manaakitanga a te mea ngaro, ki runga e tēnei, ki tēnei a tātou, kia mahia te hua maki maki hiki hiki, kia toi te kupu, toi te mana, toi te araha, toi te reo Māori, kia tūturu, kia whakamoha, kia tina, homie, hui, tai. Yeah. So, welcome everyone. That was a hui to start us on our journey. It's great, my great pleasure. Oh, just a couple of housekeeping. We're going to leave questions to the end and in the chat. So, if you have any epiphanies or questions for today, please wait to the chat at the end. But it's my great pleasure to introduce Dr. Mariana Tarallo. She's currently working at Lincoln University. She's doing some amazing mahi, and this project, the work she's going to talk to you today, is around our nemesis of Kauri. And I'm really looking forward to your talk today, Mariana. It's a good topic to have done your research on. But I'm going to leave the floor to you and do a little introduction about yourself, however you to, and welcome you to give your presentation. I'm really looking forward to it. So kia ora, welcome, no mai haramai to you and everyone here listening.
Thank you, Nick, so much for the introduction. I'll just share the screen first before starting. Is that all?
It's coming. It's coming. It's coming. Yep, there it is.
Perfect. Yeah, so thank you. Thank you, everyone, for being here. I'm very excited to be able to share a little bit about what I did in my postdoc in Mass University. Of course, a lot of the things that I'm going to show here, it wasn't just done by me. We had, it was a large group of people working on this, and it was a big team effort, but it was a project that I was very happy to be part of. I definitely learned a lot, and small spoiler alert, this is not a complete story, but I think it's a very interesting one that, again, I'm very happy to share with you guys. So to start, plants, just like us humans, they have an immune system that allows them to defend themselves against different diseases. These diseases can be caused by different pathogens like bacteria, fungi, oomycetes. And over the years, there have been many different models that try to explain how plants interact with pathogens. I'm not going to go into details about these models, but they started as simple models, and as scientists have learned more about these interactions, these models become more complex in order to accommodate all of this new knowledge. But basically, pathogens have the ability to secrete small molecules in order to facilitate infection. And these small molecules, they're called effectors. They can be small RNAs, they can be secondary metabolites, but here we're treating them as proteins. So pathogens secrete these effector proteins in order to facilitate infection. Depending on where these effectors act in the cell, they can have different names. So, for example, if the pathogen secretes an effector that works that acts in the apoplastic space, which is the space between the cells, they're called apoplastic effectors. And if they go inside the cell, they're called cytoplasmic effectors. Either way, plants also have its own special molecules, so receptors or resistance proteins that can recognize these effectors, and then this leads to the activation of defense responses. The plant can activate different defense responses. It could be activating, the induction, activating defense genes or antimicrobial compounds. It can be in the form of physical barriers, or closing the stomata, or callose deposition, so making it difficult or harder for the pathogen to penetrate. But one of the other forms of these defense responses is what we call hypersensitive response, HR, which is a form of a localized cell death So basically, it's the plant killing a small part of itself to stop pathogen growth. So if we look at this is perfect. The pathogen secretes something, the plant recognizes it, defends itself, the pathogen stops growing, there's no disease. But unfortunately, we know that's not the case. So pathogens, they also have the ability to either secrete other effectors that can escape this recognition, or effectors that are actually being recognized by the plant. But instead of actually triggering defense responses, they can suppress these defense responses. And then the pathogen growth continues. So as we can see, and as one of these models described, there's this constant arms race between plants and pathogens. So pathogens will evolve to secrete new effectors to try to avoid recognition, and then plants will always evolve to try to detect these effectors, and this goes on and on. But basically, what all these models highlights is the fact that pathogens use these effector proteins to manipulate plant immunity. And that's what we wanted to understand a little bit more in our pathosystem, that is Kauri and Phytophthora agathidicida. Kauri is one of the largest and longest living tree species in the world. As we know, it's native to New Zealand, and it has an immense cultural significance. But unfortunately, it's threatened by Kauri dieback, which is a lethal root rot pathogen disease that is caused by the pathogen Phytophthora agathidicida. Some of the symptoms include crown decline, in some extreme cases that can lead to tree mortality. There's also excessive resin production. So the pathogen causes a dysfunction in the vascular system in the plant and then leads to this excessive resin production. And Phytophthora is a soil-borne pathogen, so it means it's found in the soil and infects the plant through the roots. So that's why it's very common if we go on hikes in the North Island, in parts of the North Island, that we will see these cleaning stations. So the idea is that we're trying to stop or minimize soil movement from areas where Phytophthora could be present. So not to bring the soil into other parts of the forest where Phytophthora is not present. So in our lab, we were interested in trying to understand a little bit more about the molecular aspects of this interaction. And to help us do that, we have a few tools that make our life slightly easier, or not so hard. One of the really cool things we found is that Nicotiana benthamiana is an alternative host of Phytophthora agathidicida. So we know that Kauri is the natural host, but we found that Phytophthora could also infect this plant. And this is considered a model plant, which it's a model plant because it's a small plant, it's easy to grow, fast to grow. There's a lot of information about it. It's been used, many, many years scientists have been using these plants for many years for functional studies. So like I said, a lot of information about, the plant genes, receptors, a lot of established protocols. So that's on the host side. If we look at the pathogen side, we have a chromosome level assembly of the Phytophthora agathidicida genome. And that's important because, like I said, pathogens, including Phytophthora agathidicida, they secrete these effectors, and we can use the pathogen genome to predict these effectors using different bioinformatic tools. And so having such a good quality genome allow us to do these predictions in a more confident way. So that's what we've done. We had the Phytophthora agathidicida genome. We predicted effectors that they're potentially being, secreted by the pathogen, and then we found, all of these different numbers of effectors. So we can, like I mentioned at the beginning, we can classify depending on where they act on the cell. For this presentation, I'm going to focus on specifically two effectors. Both of them are cytoplasmic effectors, and both of them belong to a class called RXLRs. So RXLRs is a class of cytoplasmic effectors. But before we go to these two, as we can see, we have this large number, and we need to find a way to narrow this down a little bit because it's a really big number to be working in the lab, and to try to find, the function of all of this is almost impossible. And of course, because, these are predictions, right? So our prediction could be wrong, and not necessarily all of these are real effectors. So we need to find a way to narrow this down a little bit. And one of the ways that we do, especially when we're talking about effectors, one way that we have to do that is screen those effectors in the plant by overexpressing them using an agrobacterium transient transformation assay. So the idea is that Agrobacterium tumefaciens is a plant pathogen. It has some virulence genes. This bacteria has the capacity to naturally transfer these virulence genes to the plant. This gets incorporated into the plant. The plant produces the corresponding protein and then makes the plant sick. So what scientists have done is if we remove these virulence genes, we can insert then any candidate gene that we're interested in study the function, and then we make the plant produce the protein for us, and hopefully we can, work with that. So the idea here is we had all of these different agrobacterium with all of these different effectors that I showed before. We infiltrate a solution into the plant. The plant then produces the effector for us. If it's being recognized by the immune system, we would observe the cell death response that I mentioned early. So that's an indication that these proteins are, potentially being recognized by the plant immune system. So great. We've done that for a lot of those effectors that we predicted. Here, just going to focus in one of those. So we found that RXLR24 could trigger cell death. So what's happening is we infiltrate agrobacterium. Again, RXLR40 is incorporated into the plant. The plant produces RXLR40, very likely recognizes it, triggers defense responses, which is what we see here in the form of a cell death. So that was good, right? Like I said, this screening, we can just differentiate between what triggers cell death and what doesn't. For the ones that doesn't, they still could be important. So we also need to test them. We just need to keep in mind that the fact that they don't trigger cell death, it could be that, either they, like I said, is a prediction, so it could be wrong, or they are, we are testing in a specific plant, so the Bentamiana plant. So it could be that maybe this plant does not have the corresponding immune receptor, or it could be that these receptors, they are being recognized, but instead of triggering defense responses, they're actually suppressing defense responses. So we can also screen for that. So that's what we've done. And we found one specific, RXLR40, that could suppress the cell death triggered by RXLR24. So RXLR24 goes in, triggers the cell death, and then this cell death is suppressed by RXLR40. So suggesting that it's suppressing defense responses. We tested that with other cell death elisters, so we can see here we have this effector triggering cell death, the cell death then being suppressed by RXLR40. The same here, this effector triggers cell death, and then the cell death is suppressed by RXLR40. And then just to try to corroborate that, what we did was, if we overexpress RXLR40 by itself, so we know it's not going to trigger cell death. So we overexpress XLR40 on one side of the plant, on the leaf, and on the other side, a negative control. We put the pathogen on both sides, and then we can see that we have bigger lesions on the side that RXLR40 is present. So that means RXLR40 helps the pathogen, so it enhances Phytophthora infection in the plant, again, suggesting it's suppressing defense responses somehow. For this bottom part, we don't need to worry so much, but what we did is just another way to try to corroborate that, but also to have a sort of a negative control for any future experiments. So this is what our XLR40 looks like. So that's the sequence. So here it has all the information that it needs to be able to to have the suppression activity. What we did is just a few changes in order for the suppression activity to be abolished. So if we look here, when we infiltrate something that we know triggers cell death, and we infiltrated this RXLR40 mutant, the cell death is there. So it's not suppressing the cell death anymore, and it is also not helping the pathogen anymore. So again, it's just to show that RXLR40 has this activity, and also for to serve as a control for some of the other experiments that I'm going to show. So cool. We have RXLR40 suggesting that it's suppressing defense responses. So what happens to the pathogen when we remove this gene? And we did that by RNA interference. So RNAi is a natural defense mechanism that is present in us humans, in plants, in pathogens, and is a mechanism that cells use to switch off specific genes. So if we think about us, we have thousands and thousands of genes. These genes cannot be all expressed at the same time. We couldn't cope with that. So depending on our environment, if we are sick, for example, our cells have a way to switch genes on and off. And one of the ways that it has to switch them off is through RNAi. So again, we don't need to worry so much about all of these proteins and what's happening, but I think the main thing here is to know that what triggers RNAi is a molecule called double-stranded RNA. So double-stranded RNA will always match the target gene, so the gene that we want to silence. And when it matches to that, it signals to the cell that this gene needs to be degraded. So it gets chopped up. And then the gene is silenced, so the corresponding protein is not produced anymore. So in our case, what we did was we created a double-stranded RNA that matches part of our XLR40. And we apply that to the pathogen in the hopes that, this gets taken up by the pathogen, and then this uses Phytophthora agathidicida own mechanism to silence the gene. So pretty much we are tricking the pathogen to silence its own gene. And then we can test whether, we'll see what the effect removing or silencing this gene will have for infection. So, like I said, this double-stranded RNA molecule is it's what starts it all. So we needed to find a way to be able to deliver that to the pathogen, but also be able to test if it was going to have any impact on lesion. So the way that we did that by two different ways. So in our model plant in Nicotiana, what we did was we spray a solution of double-stranded RNA that had this fragment that matched XLR40 into the leaves, and then we put the pathogen, and then hopefully it will get taken up by the pathogen and we will silence the gene. Will this double-stranded molecule also be taken up by the plant? Very likely, but we need to remember that this double-stranded RNA it only matches with our XLR40, and our XLR40 is only present in the pathogen. So even if it does go into the plant cell, it's not going to match anything because there's nothing similar to our XLR40 in the plant, so nothing will happen. So it will only silence things on the pathogen side. For Kauri, we did something slightly different because how different the leaves were. So instead of spraying, what we did was to infiltrate the double-stranded RNA solution, and again, just applying the pathogen, and the idea is the same. The pathogen will take up the double-stranded RNA. So once we figured out how to deliver these molecules, the next step was to see if it was actually silencing XLR40. And here we can see that it does. So we have lower levels of our XLR40 in both of their hosts. So it means that our XLR40 is being silenced. So from that, the next question was, okay, if the gene is silenced, what is actually doing to the infection? And we can see that it also can reduce the infection. So on the leaves that our XLR40 are silenced, we can see lower lesions. So it's reducing Phytophthora agathidicida infection in both of our hosts. So that's really cool. So all of that together, with that previous slide shows that XLR40 seems to be a very important effector for Phytophthora agathidicida, and it's somehow suppressing defense responses. So our next question was, okay, XLR40 goes in. It's very likely interacting with something in the plant in order for the suppression to happen. So we wanted to figure it out what was interacting in the plant for the suppression to happen. And we did that by a technique called yeast to hybrid. So as the name suggests, all of this happens inside a yeast cell. So what we do is we add XLR40 inside a yeast cell together with a library that has many, many, many plant genes. So basically what XLR does is that it looks for its perfect match. And then once XLR40 finds a protein that it likes, that it matches, this physical interaction then allows the expression of some genes that allows the yeast to grow in selective media. So we found that XLR40 interacts with ARIA. So we can see here what a positive interaction looks like. So RXLR40 interacts with ARIA, so that's why we see the yeast growing in the selective media. This is just a positive control, so everything will grow. And this is what a negative interaction looks like. So when we put RXLR40 mutant, so that one that I've mentioned that does not have any suppression activity and does not enhance pathogen infection, when you put together with ARIA, we don't see the yeast growing because they are not physically interacting. And yeah, so what is this protein, right? So ARIA stands for arm repeating protein interacting with ABF2. ABF2 is a transcription factor that is part of the abscisic acid signaling pathway, so it's a plant hormone. And then when ARIA was first identified, it was then described as being a new component of this pathway. So that was nice because it gave us a small hint of what RXLR40 might be, targeting or manipulating in the plant. Here we have another way of showing that the interaction happens, but now it happens inside the plant cell. So all of this, the interaction is happening inside the yeast. So we just need to prove that the interaction actually happens in the plant, where it will be happening in real life, let's say. And also we wanted to find out where exactly these proteins localize. So once in the case, once RXLR40 goes in, we had an idea that it was cytoplasmic, that goes inside the cell, but then we also saw that it goes inside the cell, but also goes inside the nucleus. And that's where ARIA is, so it makes sense. So that's where the interaction happens. So the interaction between RXLR40 and ARIA happens in the plant nucleus. The other thing that we wanted to test, based on the results that we had with RXLR40, was if its interacting partner ARIA could also suppress the cell death triggered by the previous effectors that we had tested. So we can see here, the effectors like that I mentioned in one of the first slides, they are triggering cell death, and then when we add ARIA to it, the cell death is then suppressed. And then similar to with a virulence assay. So when we overexpress ARIA by itself and we add the pathogen to one side, negative control on the other, we see larger lesions of the pathogen. So, all of this suggests that ARIA might be behaving as a susceptibility protein. So like I said, ARIA is a component of the ABA signaling pathway. So we wanted to see what effect would ABA have on Phytophthora infection. And this is because there are many studies that have shown that, in some cases, ABA help pathogens to infect plants. In other cases, it has been shown that ABA helps the plant to defend themselves against pathogens. So we knew RXLR40 interacts with ARIA, but is this interaction, is it somehow making ABA levels go up or go down? And either way, is this helping or not the pathogen? So what we see is that when we spray ABA into the leaf, so here we don't have any overexpression, it's just the leaf. We spray ABA, we put the pathogen on each side, and we see that it helps the pathogen quite a lot, so it enhances infection. And here, on this other side, when we overexpress ARIA, we see that there's an induction of some genes that are involved in the biosynthesis of ABA. So this all kind of suggests that RXLR40 somehow might be hijacking this hormone pathway to increase, or at least to try to make maybe ARIA increase genes that will lead to the activation or the increased levels of ABA. So one of the last things we've done was to see what ARIA interacts in the plant when RXLR40 or when Phytophthora is not present, right? So we know RXLR40 interacts with ARIA, but when ARIA is in the plant without the presence of the pathogen, what is it interacting with in the plant? And then we found that interacts with a next transcription factor that is called SOG1. So SOG1 is involved in DNA damage response and oxidative stress signaling. So all of this is consistent with triggering defense responses. And that's what we see here, right? So when we overexpress SOG1 on one side, the negative control on the other, we see that we have smaller lesions compared to the control, which is the opposite of what we saw for RXLR40 and ARIA. So it's very likely activating defense responses somehow. So this is the part where I've mentioned that unfortunately we don't have a complete story yet. I think we definitely know a little bit more than we did when we first started, but there's definitely a few things that we would be really nice to understand more. I think especially related to this interaction here when the pathogen is not present. So if SOG1 is actually a resistance protein, what exactly is it doing to ARIA? Does it bind so then ARIA is not free in order to induce the expression of genes that will lead to the increase of ABA levels? And then when RXLR40 is present, um, what does it do to this interaction? So is it competing somehow? Or does it compete in a way that so then ARIA doesn't interact with SOC1 anymore, and then it kind of frees ARIA to increase ABA levels? Are there other proteins that are involved that maybe somehow maybe can target ARIA? Sorry, that can target SOC1 for degradation. So those are things that we don't know yet. And like I said, and showed at the beginning, Phytophthora and many other pathogens, but Phytophthora secretes hundreds of effectors, and these effectors many of them will be acting in different plant pathways. Some of them could also be acting in like hormone pathways. So this is just a very, very small part of a very complex interaction between this pathogen and its host. But I think it just highlights the fact that how important it is to understand more about these effectors, understand what they're interacting or affecting in the plant or the pathways that they are affecting, and hopefully this can lead to better control strategies. But I guess that even if that doesn't happen, just the fact that we can understand a little bit more and move on a little bit from the pathogen secretes this effector and they suppress defense responses. So just the fact that trying to answer how exactly these defense responses are being suppressed, I think it's really interesting and really valuable as well. So yeah, that's me. I would like I said, there was a lot of people involved in this project, but I would like to thank everyone in Massey, especially Rosie and Carl. They were my PhD supervisors. They also helped me quite a lot in this project. Also, this project was completed under the authority of the Aotearoa iwi, and they also very kindly provided us the Kauri seedlings for not only this project, but other projects that we have. Also, I've done part of my research in the James Hutton Institute in Scotland as part of a collaboration that we had with them. And of course, by protection for all the support and you guys for listening. Thank you.
Oh, my te paki. Thank you, Mariana. That was amazing. And fantastic that you had the iwi te roroa involved with your mahi or your research as well, and acknowledging your wonderful supervisors too. So, any chats or any questions, Patai, please put them in the chat. In the meantime, I've just put the link there that your presentation that you've just seen today, if you want to refer to it, anyone. It will be at that link. It won't be yet, but it'll be uploaded shortly. And reminding everyone here that all previous Rangahau Rising webinars, including Sarah Herbert's one on myrtle rust last month, are all available at that link, as will your fantastic presentation. So please do put any comments or questions in the chat. Kia ora, Kate. Very interesting work and great use of classical molecular genetic tools. Well done. So there you go. Great presentation. I was intrigued by the Kauri leaves are notoriously hard to work with because of their thick, waxy cuticle. So well done for also being able to navigate that, I guess, inherent constraint often to some research with Kauri. Just thinking about your presentation and your work research overall, what do you think some of the challenges and possibilities for delivering, let's say, double-stranded RNA tools in the field, and as a strategy to control Kauri dieback, what do you think those challenges and possibilities will be for such a
I think
novel
there's
and tool? Yeah.
definitely a lot of challenges. This this double-stranded RNA, it's a molecule that it's very, unstable. So definitely how long it would last in the field, it's would definitely be a problem. Uh, Also, I mean, it's not as expensive as it was, but it's still quite expensive to produce them. So that's also another issue. But I think, yeah, how long they would last, and they're, they can be degraded by UV light and rain, or even other microorganisms that, are going to be around. And definitely how this would be applied, right? I don't think it's definitely something that should be done in the nursery. I don't think, unfortunately, it's something we can just go inject that in massive trees, and that
Yeah.
would work. But yeah, there's some studies that show that there's some delivery of double-stranded RNA through the soil, and then it gets taken up by the plant, and that
Ah,
protects them. But yeah, it's just very, very early on. And so I think there's definitely a lot of challenges. I think especially with not only how it will be delivered, but how to make sure that, or also like how many times you have to apply, but I think mainly related to the fact that these are very unstable and degradable molecule.
is there, in your knowledge, is there work on stabilizing it as a control tool, like in other parts of the
I
world
think
or
there's
other
some studies that try
programs?
to how they can deliver that using some particles or things like that.
Yeah,
But I don't think it's something that it's very like I don't know with like we see with phosphite for Phytophthora, right? It's not something that it's as sim not that doing that it's simple, but just yeah, definitely it's not as it would be way harder to do that with these molecules, but
yeah. But I mean, having a toolbox, because there's the limitation for coated dieback management in terms of infected trees, rako, or affected, infected ecosystems, forests. It is very hard to have a one-trick pony, so to speak. Well, it has been. So developing tools such as this would actually be, expanding the situations and applicability of coated dieback management, but also, I guess, other phytophthora management systems
yeah.
too. So, I mean, difficulty shouldn't be as it might be a challenge, but it would be good to see that even these areas are expanded. A question from Polly. Thank you. Very interesting work. Kia ora, Polly. I'm curious about how you narrowed down to RLX 40 and RLX 24 and why you checked them together. Also, are those effectors present in other Phytophthora species?
Yeah, so I think that it seems very simple to go from one slide to the other where, we had like 150 factors and then we screen all of those and then we found this magical two. But this part of the work, it wasn't done by me, it was done by a previous postdoc. So she cloned a lot of these effectors and she screened a lot of those effectors. So that's how we could, so from this 100 and something, we then separate those that triggered cell death, and then which I don't remember exactly, maybe I think we're around 8 or 10. And then from the ones that didn't, she also tested to see if they could suppress. So there was definitely like a lot of work involved. So it wasn't just like magically finding these two. Um, and yeah, that's a good question. So RXLR24 is actually present in other Phytophthora species, but RXLR40 seems to be present only in Phytophthora agathidicida, which is interesting, right? That we couldn't find any homologs in any other Phytophthora
Oh,
species. So it seems to be very specific.
that's very
And interesting that it seems to be targeting a very, very conserved pathway, which is, hormone pathway. It's the ABA pathway. It's quite conserved across many, many plant species. So it's interesting. At least we couldn't find based on their sequence. So we couldn't find any homologs, but there's always the possibility that, their structure, the protein, the 3D structure. So as we know, like the protein will fold in order from like its 3D structure, which then determines the function. So it could be that if we looked into that, we would find similar homologs in other Phytophthora species. And just to, I think something I didn't mention is that so Artsalar is a class of effectors that it's only present in oomycetes, which
Oh,
like
right.
Phytophthora. So fungi, for example, they don't have these effectors, this class of effectors. Of course, they will have different that could even interfere with hormone pathways like that, but not specifically this class is only present in Phytophthora.
Oh, so that was part of the question then of how that was narrowed down, and that's one of the reasons. Yeah, yeah, no. But there may, would you think, do we know if there are other types? It
Of
was just been that's been identified as those are the ones we should be targeting for oomycetes, or is that
There's
still?
definitely like in one of the pictures that I've showed, one of the effectors that were suppressed by RXLR40. So it was
Yeah,
also part of another student PhD project. So she found another. So we've been like screening and working a little bit with this effector. So yeah, there's definitely others that triggered cell death. I think this is the only one that we found that could suppress the cell that it's only present in agathidicida, but definitely there's definitely other things that we can look into. That's why I said it's just a small part of a bigger story. So Phytophthora was secreting many other things. Um, so
but no, it's been great to get that mahi done, particularly for those initiatives being happening in Tranche 1 at Massey. So kia ora to you and others. And thanks, Polly. Some good, awesome questions. Um, any others? Just keep them going in the chat. In the meantime, what do you think, I guess, the limitations would be, say, for using the Nkotiana model system to study, let's say, a Kauri pathogen? Is that something that you feel is a good, well, there may be limitations, are there, or not?
oh, definitely yes, because it's not. So you're doing all this study in a plant that it's not the natural host. And again, so Cody is a gymnosperm, Nicotiana is an angiosperm. So they're very, very different.
Yeah.
So there could also be components, that's not present in one that is present in the other. So that would make things, slightly different. We do have, hopefully soon, a paper coming up where we looked into the expression of Phytophthora genes when it's infecting Cody, when it's infecting Nicotiana benthamiana. We do see a lot of similarities, so I think it is definitely an alternative host, but it's not the natural host. And I think something that we definitely see it's a difference in the time of infection. So the infection in Benti happens way faster than it does in Cody as well. In Cody, so we just need to keep in mind that when unfortunately we cannot like generalize things um, and I think just again, just very that just shows the importance of validating this. We can use these model plans; they're very valuable to discover things. But I think we need to try to validate somehow in the actual host. But yeah, especially with forest trees, it's something that it's very tricky.
Yeah. No, kia ora for that answer. Um, Kia ora Maria. She has a question. Again, very good presentation. Do you think this framework could also be applied to assess whether Kauri is showing stronger resistant traits to PA infection demonstrate improved response outcomes?
You mean the RNAi thing?
Yes, I think that's what she means. Yes, the framework being the
I think this would be something. I don't think we could. Well, I mean, I think we could. I think it's just a matter of, this would, I think this is something that would happen in nurseries before, the plant has any contact with the pathogen. I don't think we could do that, if the tree is infected, but I'm not sure. This is just very early research, but I guess, yeah, I don't think, I think,
yes, using the molecular pathways, yeah, just to refine that.
Yeah, I think for this kind of work, we definitely wanted a genotype of Cody that was more susceptible because in this case, we actually did want the pathogen to infect like easily so we could, see if we saw any difference. But maybe, yeah, it could be something that could be applied for other genotypes that have that show resistance or more tolerance to Phytophthora. Maybe try to
Yes, there have.
understand why that's the case or why some of them.
Mm. And that work, it would be quite good because that work began with the Healthy Trees, Healthy Future program at Scion many years ago. And there was the PA was such an aggressive pathogen on a lot of the Kauri populations, I guess, that were tried. It was very hard to, but there had been some tolerance or resistance indicated. So I guess this could be a good way of if that work was to continue at any point, which I think it should, of course, this might be a good way of helping that progress. I mean, I think it would, but yeah,
I know,
just
definitely.
seeing what your results so it would be a good way to do that. Kia ora, Lisa. Lisa, nice to see you here today. Prophylactic defense barrier of sorts with a buffer of planted resistant trees. Yeah, yeah, that would be
Oh, yeah. I don't know exactly how. I think we have different levels of tolerance, but I don't know if there's a resistant, completely resistant
no,
genotype, is there?
no. Well, that if there is, it hasn't been truly confirmed or identified to my knowledge, but it may. Yeah, I don't. Yeah, I guess the that's the point of looking and having the methods to look
Mhm.
because yeah, I guess demonstrating absolute robust genetic resistance would be just it'd be quite hard to do without the sorts of methods that are being developed here. Um, also, yes, but yeah, kia ora. That's yes, opportunities for lots more research here, clearly.
Mhm.
Was there anything else you wanted to add as a sort of an afterthought, or what would a key area you would like from your work to see explored further?
Well, I really like this fundamental part of things. So I think, continue, like I said, maybe it will not lead to, I don't know, a 100% control strategy, but I think just the fact that we can understand a little bit more, I think that always brings a lot of, there's a lot of value in that. So I would love to, if I had the opportunity to continue this project, with trying to continue to understand this small part of this interaction. I think it's very interesting.
Mm. And I think I think, as we have a lot of like catastrophic declines in various ecosystems and keystone species like Kauri, I think there is a need to explore many long-term solutions and ones that are less, let's say, yeah, especially the ecosystem engineering that happens with a long-lived species like Kauri, average age between 500 and 1,000 years. It's got that longevity really needs a solution that provides that very long-term, I guess, protection or response to something like a catastrophic disease such as agathidicida. So I think that's where some of these longer term approaches, although they're very hard to get funded because everything is in short term cycles, having that longer term outlook, I think, for a system such as a Cody system, which is such, A, a native ecosystem, but B, a very long lived one, I can't see how, I mean, at the moment, those sorts of methods that you are working in is going to be providing the fundamental research provides some opportunities to then get that longer term outlook, which I think has been missing with a lot of our, I guess, pest and disease management. It's all very short term or around a crop, cycle of production. But how do you protect a long-lived ecosystem? I guess that was why was more of a comment and why I think and totally endorse tautoko your support that for around this longer term fundamental research is absolutely absolutely vital. So just really endorsing the mahi you and your esteemed colleagues, kia ora Carl, Rosie and others have shown us and provided us for those insights. So thank you again. Um, any last questions? Otherwise, I will just remind everyone that we have a Rangahau Rising next month, July 15th. And it's going to be from Dr. Celine Schloffel Armstrong, and the title is "Are We Still Repeating the Past? Tracing the Norms of Biosecurity Across Time on Te Pātaka or Rākauhautu or Banks Peninsula." So the webinar will be focusing on how pests and weeds first emerged and as threats to the land and agriculture in Aotearoa. So doing a sort of a time scale. So if you'd like to, please do join us next month for that webinar. It's very register as you did for this one. And reminding that Mariana's fabulous presentation will be at the Rangahau Rising seminar series. So thank you to all of you. Ah, there we go. There's the link. There's the title. Um, just thank you for joining us today for today's corridor. Thank you, everyone. Looking forward to next month with Celine, and thank you again, Mariana, for your mahi today. Um, I'll just close us off for today and whakamutunga karakia and send you on your afternoons. E te papatuanuku kua motu tātou e te mahitahi o te rā, kia tātou rangamārie ke mātou. Te hei Māori ora. Take care everyone and see you next month. Ka kite anō.